Module 21. Now, let's talk a little bit about gastric motility. One event occurring with the stomach as it's moving around is something called the receptive relaxation response. Mechanoreceptors in your pharynx send signals to the medulla oblongata as you are actively swallowing a bolus. The medulla oblongata then sends signals to your stomach. In response to those signals, your stomach will resist stretching, but only briefly. It then almost immediately relaxes to accommodate the arriving food. Once the food has entered your stomach, there are mixing waves that are going to occur that are going to help to macerate your food, mix it with your gastric juice, and form your chyme. You're then going to exhibit in the pyloric region of the stomach gastric emptying. Now, these are mixing waves that are going to periodically force 3 milliliters of chyme at a time through the pyloric sphincter and into your duodenum. This very slow emptying of the stomach enables your duodenum to neutralize your stomach acid and digest your nutrients little by little. Now, from an average size meal, it takes about 4 hours for your stomach to completely empty. Now, less time might be required if your meal was more liquid. More time is needed if your contents are acidic. And high-fat meals can take up to 6 hours to empty, which is why you tend to feel fuller longer after having a high-fat meal versus a meal that was more high in carbohydrates or high in protein. It's estimated a high carbohydrate meal can empty in as little as 2 hours. High protein meals more along 3 to 4 hours. Now, if you ask yourself, can your stomach absorb nutrients? Nutrients are things like monosaccharides, amino acids, vitamins, those types of things. The answer is no. And there are several reasons for why. One, the epithelium is covered by a really thick blanket of mucus. And in order to absorb nutrients, the nutrients have to contact the surface of the epithelium. So if you've got all of this mucus, it's basically forming a barrier between the nutrients and the wall. Also, even if nutrients could contact the lining, the surface of the epithelium itself lacks the transport mechanisms needed in order to absorb the nutrients. So those mechanisms you find in the small intestine to absorb nutrients aren't here. The lining of your stomach is almost even impermeable to water. Almost, but pretty close. And besides, at this point, digestion's not even completed. When I talked about salivary amylase in the mouth, I said it begins the digestion of those big polysaccharides, but you're not done. The maltose and the oligosaccharides are still too big to be absorbed. And the pepsin begins the digestion of proteins into peptides, but that peptides are still too big to be absorbed. Now, by saying the stomach can't absorb nutrients does not mean that it can't absorb anything. There are some things that it can absorb. It can absorb a really small amount of water and alcohol. Now, the intoxicating effects of alcohol really depend on how long it takes for your stomach to empty. The faster your stomach can empty, the faster the alcohol can get absorbed by the small intestine. You can absorb some ions across the epithelium of the stomach, short-chain fatty acids, Short chain means that the hydrocarbon chain does not have that many carbon atoms in it, so it's just physically a shorter molecule. Also, you can absorb some lipid-soluble drugs here. Most of your pain medications are lipid-soluble, so most of your pain meds can be absorbed across the lining of the stomach into the blood, which is going to allow you faster relief from pain. Aspirin, when you've got that migraine and you need to get that pain relief, getting some aspirin into your system as quickly as possible, definitely going to be important. Now, because your stomach contents are so corrosive, there are some protections in place to help protect the wall of the stomach from damage. You have that lovely mucus coat. Mucus is alkaline, so it does resist the actions of the acids and enzymes by forming an alkaline barrier. It also is a physical barrier, keeping those acidic contents away from the wall of the stomach. The cells forming the epithelium are joined by tight junctions, which, and remember from A&P1, in Chapter 5, when you learned about the histology, tight junctions basically seal the spaces between cells. And it's going to limit the movement of materials across that epithelium. So think of the tight junctions as a giant Ziploc seal, and by having these junctions in the epithelium of the stomach, you have ziplocked that entire surface, and it keeps your gastric juices from seeping in between the cells. You also have a lot of epithelial cell replacement. On average, epithelial cells only live between 3 to 6 days, and once they become damaged and old, they slough off into your gastric juice, but they're immediately replaced by new epithelial cells thanks to those regenerative cells in your gastric glands. Now, there is one notable major event that involves your stomach, and it's called emesis. Now, the emesis is the fancy biological term. Vomiting is the more commonly used term. Now, when you vomit, when you undergo emesis, you have the forceful expulsion of the contents in the upper GI tract out through the mouth. Now, the upper GI tract would include the stomach, but also the duodenum, which is the first portion of your small intestine. Now, the act of vomiting does not involve the muscle in the walls of either of these structures. What's going to happen is you're going to violently squeeze your stomach and your duodenum in some cases between your abdominal muscles and your diaphragm. So your diaphragm and your abdominal muscles are going to contract with great force, compressing the stomach and your duodenum between them. And by applying that external force, you basically push contents up through the lower esophageal sphincter, up through the esophagus and pharynx, and out the mouth. Which is why, after having, let's say, stomach flu, right, to use the common phrasing, after you have recovered and you're no longer vomiting, it actually feels like you've done way too many crunches. There are some people that you actually feel like you have over-exercised the muscles of the abdomen and you're sore for a couple of days. Think about it this way. It's a great ab workout. The more you vomit, the more violent those contractions happen. It's great exercise for those muscles, though it's a pretty unpleasant event. Now, the vomiting reflex is controlled by the vomiting center in your medulla oblongata, and this center has varying triggers from person to person. There's different people are more sensitive to different triggers. In fact, this is a really huge area of ongoing research in the neuroscience community. In fact, at the annual Society for Neuroscience meeting, they actually have a brunch for the Emesis Society. Anyone can go to it. My friend Leanne in grad school, and then when she did her postdoc work, she made sure she went to their brunch every year. Apparently, they have great food, and it's a 4-hour seminar just talking about the new findings in vomiting research. So, and honestly, the articles are fantastic to read. A little disgusting, depending on your personal preference, but really fascinating. Now, some of the different triggers that could make this vomiting center initiate the sequence of events for vomiting would be unpleasant sights. There are people that will throw up at the sight of blood, so just that visual presence is enough. There are people who can trigger this through smell. In fact, the girl that I dissected my cadaver with when I did human growths in grad school, the moment you would open up the gurney to do the dissection, that smell of formaldehyde would hit her, the embalming fluid that they used, and she would throw up. We had lab twice a week, and she would throw up right at the beginning of lab every single class, so they just had a trash can waiting for her, and it just sort of became this accepted thing. It was going to happen. She would throw up once, and then she'd be fine. But it was that sudden exposure to that smell for her triggered this vomiting reflex. Stomach irritation, which could occur with an infection, so a good viral infection. Also, excessive stretching. Have you ever eaten so much food that you just felt sick afterwards? And you just kind of felt like, oh, I'm so uncomfortable, I feel sick. That is your stomach stretched beyond what it should be, and it's actually knocking on the vomiting center door. So you're on the verge of actually triggering that reflex, because if your stomach has stretched too much, there's a risk of potential damage, and your vomiting center could trigger you to vomit, which would empty out the extra contents and protect your stomach. Dizziness can cause vomiting. So that's why amusement park rides where you're doing lots of spinning, people get off the rides, vomiting can occur. Various types of drugs like morphine, so a lot of the pain medications in some people can trigger vomiting to occur. So from person to person, what trigger is going to actually cause you to vomit will vary. All right, so what I'd like to do is leave your digestive tract, because we have now gone through your stomach. We are emptying things into your small intestine, but I want to talk about your accessories now that are in the abdominal cavity, because these accessories are going to empty secretions into your small intestine. And the first accessory I want to mention is your pancreas. Now, the pancreas was mentioned in your endocrine chapter because it produced things like insulin and glucagon. What we're going to talk about here are the exocrine functions, the digestive functions. Now, about 99% of your pancreas is composed of structure called acini or pancreatic acini. These acini are going to produce about 1.2 to 1.5 liters of pancreatic juice per day, and this juice is going to be carried into the duodenum, the first section of your small intestine. Now, this juice is mostly water, but it's got some really interesting enzymes in it that are essential for the breakdown of your nutrients. For example, there is pancreatic amylase, which is identical to salivary amylase, except it's made by the pancreas, not a salivary gland. So pancreatic amylase is going to take your starches and your glycogen, those big polysaccharides, and it's going to continue breaking them down into oligosaccharides and maltose. You then have trypsin and chymotrypsin, which are going to take your big polypeptides and start chopping them up into smaller oligopeptides. You have carboxypeptidase, which is going to take your oligopeptides and chop them up into amino acids and some smaller peptides. You have pancreatic lipase, which is your primary lipid digesting enzyme. So this is going to take care of the 85 to 90% of your triglyceride digestion down into fatty acids and monoglycerides. And interesting to note, fatty acids and monoglycerides are small enough to absorb. So this is a one-stop shopping. You have one major enzyme, it breaks your triglycerides down into absorbable nutrients. Now, when people think of nutrients, they think of proteins and carbs and they think of fats. They don't think about DNA and RNA. And when you eat plant products, when you eat animal products, you're not just eating their proteins, their starches, their glycogen, their fats, you're eating their DNA and RNA also. And your body has those components. So the DNA and RNA of your plant and animal foods can be broken down into building blocks you can use. So you have in your pancreatic juice something called ribonuclease. Ribonuclease is going to break your RNA down into your nucleotides. Deoxyribonuclease breaks down your DNA into nucleotides as well, so it breaks them down into those building blocks. However, nucleotides are still too big to absorb, so their digestion is not quite done. The last component of pancreatic juice I want to mention is sodium bicarbonate. Now, sodium bicarbonate is not an enzyme, but it does help to make your pancreatic juice alkaline in pH. And this alkaline pH is going to help to buffer the acid in your chyme as it is leaving the stomach and coming into the duodenum. So this neutralizing of the chyme is going to protect your duodenum from acid damage. The alkaline pH also inactivates pepsin, because pepsin only works at an acidic pH. And all of those enzymes coming from the pancreas have to be in an alkaline environment to work. So you have to have a nice alkaline pH in the duodenum and the rest of the small intestine for any further digestion to occur. Now, we're going to talk a little bit about your liver and your gallbladder. Now, the liver is your second largest organ in the body, but it is your largest gland. So if you're playing Trivial Pursuit and you get a question, what is the largest organ in the body? Please make sure you remember to say skin. Your skin is your largest organ, liver is number 2. But if the question said, what is your largest gland? Liver is your answer. Now, as I mentioned in our first module, all the varying functions of the liver are independent reading. However, I do want to talk about the function of your liver involved in this system, which is the production of bile. Now, before I start talking about bile and what it does, I do want to mention your gallbladder briefly. Your gallbladder is on the inferior surface of the right lobe of the liver. It's a pear-shaped sac that actually stores and concentrates the bile produced by your liver until it's actually needed in your small intestine. Now, this is where you will form things called gallstones. So if you've ever had someone who had a gallstone, it's being produced in the gallbladder. Gallstones are crystals of cholesterol and mineral salts, and they're due to having either excess cholesterol in your bile, and this really high level of cholesterol will spontaneously start to form crystals, or your bile is getting too concentrated. So that means you're decreasing the water content of your bile, which is making your cholesterol concentration higher, and you're forming these crystals spontaneously. Now, crystals are sharp. Okay? They have sharp edges. So gallstones can be rather painful. They will scratch up the inside of the gallbladder. Also, you do run the risk of these gallstones flowing out of your liver, your gallbladder, and into the varying ducts that are draining bile down into the duodenum. And you run the risk of one of those gallstones actually lodging in one of those ducts and blocking bile flow. So gallstones can present some pretty serious medical problems down the road. Now, let's talk a little bit about bile. The hepatocytes, the functional cells of the liver, produce about 0.5 to 1 liter of bile per day, and this bile is going to be secreted into your duodenum. Now, bile itself is actually composed of mostly water with some solutes like bile salts, cholesterol, phospholipids, some neutral fats, bile pigments. In fact, you've learned the most common bile pigment, that lovely bilirubin, that waste product from your red blood cell breakdown, and various ions. Now, with regards to digestion, it's the bile salts that are significant. Bile salts are actually produced from cholesterol. Now, bile salts are going to perform what is called emulsification of your dietary lipids. And emulsification is going to have two steps to it. One, you're going to have to take your large lipid globules and break them down into smaller lipid globules. So you take a big chunk of fat, you break it up into smaller chunks of fat. What that's going to do is increase the overall surface area of your lipids, which is going to allow for better digestion with pancreatic lipase. The greater your surface, the more surface you have to actually break things down. Also, your pancreatic lipase is water soluble. So the pancreatic lipase is in your pancreatic juice. It's just dissolved in the watery contents of your duodenum. The problem is that lipids do not like water. Lipids are hydrophobic. So in order to digest the lipids, you have to get the lipids to mix with the water so that the pancreatic lipase can actually come up to the surface of the lipid. So part of emulsification is to get these little lipid globules, these little lipid droplets, suspended in the chyme, suspended in the water. And how this will work is you are going to take these bile salts, which are amphipathic. Now that's a fancy term that came up in A&P 1 when you learned about phospholipids. Amphipathic means one side of the lipid loves the water, meaning it's hydrophilic. One side of the lipid is hydrophobic, meaning it hates the water. So in the little diagram that I so technically drew in PowerPoint, for you, the purple thing in the middle is a lipid glob. The blue structures that look like a stick and a ball, those are individual bile salts. Now the long stick part is the hydrophobic part, and that the hydrophobic part is going to attach to the lipid glob, all around it, completely surrounding the glob. The little balls on the stick, those are the hydrophilic heads. Now, when you start putting the bile salts with the lipids, it's basically going to form a little water-loving sphere around that glob, where you've got all of these little I love the water heads facing the water, and everything that hates the water, the lipid glob and the stick part, are hidden on the inside. And because the surface of the sphere loves the water, it will mix in the water of your chyme. So emulsification is accomplishing two things. It's increasing the surface area of your lipids, and it's making your lipids water soluble. It's helping to get them into the water, suspended in the chyme, so that they have the ability to interact with the pancreatic lipase. Now, there is a role for the bile salts later on with the absorption of lipids after digestion is complete. But I'm going to save that for when we talk about absorption in our third module. Now, 80% of your bile salts are going to be reabsorbed in the small intestine, taken back to your liver, and reused to make new bile. So it is a mostly recycled product. All right, back to your digestive tract. Let's talk a little bit about that small intestine. Now, the small intestine's major functions are to receive chyme from your stomach and receive secretions from the liver and pancreas. Your small intestine is a very mobile structure, so it is going to have those segmentation movements. So there is going to be mechanical breakdown of your nutrients in here. It also is where you have your pancreatic enzymes. You also have intestinal enzymes in here, so we're going to have a lot of chemical breakdown of your nutrients. In fact, your small intestine completes all of your digestion and all of your nutrient absorption. So it's a very busy structure. Now, the small intestine does produce its own juice, produces about 1 to 2 liters of what's called intestinal juice per day. It is mostly water, but it does have some mucus in it, and it is slightly alkaline. So part of the job of this juice is to keep your chyme nice and moist, always remembering that it's going to be easier for a liquid to pass through all of the small intestine and large intestine than something that's more viscous, right? So we need it watery. The mucus is creating that nice alkaline pH, which is going to help buffer the acids coming from the stomach. And by guaranteeing that everything stays nice and wet, it keeps your digestive enzymes and the products of digestion in solution, which is going to help get the enzymes mixed up with all of the different food products that are in there, allowing digestion to continue normally. Now, the lining of your small intestine is a simple columnar epithelium, just like in your stomach. And if you look at the figure, you can see that you've got these little finger-like structures sort of on the surface of your small intestine. These little finger-like structures are your intestinal villi. Okay, a villus is a finger-like projection. And you can see your epithelium running up and along the finger that has been cut in cross-section. And you can even see that epithelium dip down and form a intestinal gland as well. Now, on the outer surface of the simple columnar epithelium, so we're on the very surface, you have what are called microvilli. Microvilli are little finger-like projections of plasma membrane along the surface of a cell. And these microvilli actually create the appearance of a bristle brush. Yeah, I don't know if everyone has seen these. They're very common up north. Bristle brushes can be used to scrape mud off of your boots before you go in a house. They're very coarse-haired brushes. Someone somewhere microscopically saw these villi, these microvilli, on the surface of the epithelium and said, wow, that looks like a bristle brush. We're going to call this the brush border. And in this brush border, you have what are called brush border enzymes. So these are just enzymes on the surface of your absorptive cells. There are a lot of different brush border enzymes. We're just talking about the major ones. You have dextranase and glucoamylase. These two enzymes break your oligosaccharides down into glucose. You have maltase, which breaks maltose, the unusable disaccharide, into the monosaccharide glucose. You have sucrase, which breaks down the disaccharide sucrose, which cannot be absorbed, into glucose and fructose, monosaccharides that can be absorbed. You've got lactase, which breaks down lactose, the disaccharide that you cannot absorb, breaking it down into glucose and galactose, monosaccharides that are absorbable. So these first 5 enzymes basically finish your carbohydrate digestion. They create monosaccharides that can be absorbed. You then have nucleosidase and phosphatase. These enzymes are going to attack your nucleotides, the building blocks of your DNA and RNA, and it's going to break them down into your nitrogenous bases, your phosphate ions, and your pentose sugars, all 3 of which completely absorbable as nutrients. So these enzymes finish the digestion of your RNA and DNA. You have enteropeptidase, which doesn't digest any nutrients, but it's going to activate a chemical called trypsinogen. Trypsinogen is made by the pancreas, and it's in your pancreatic juice. And when trypsinogen enters the duodenum, enteropeptidase changes it into the active enzyme trypsin. It's kind of interesting. Trypsin could actually digest the pancreas if it was actually made as trypsin in the pancreas. Trypsinogen, on the other hand, doesn't do anything. So your pancreas makes a chemical that is safe for itself and then does not activate until it's in the duodenum where it's safe for it to be active. Lastly, in your intestinal juice, or pardon me, in the brush border, you've got enteropeptidase and dipeptidase. These are going to finish your protein digestion. They're going to take any oligopeptides that are remaining and chop them down into amino acids, which are absorbable nutrients. Now again, the enzymes that I'm hitting here are the major enzymes. New research keeps finding more. There are more considered to be minor enzymes, but even the cells that get sloughed off the epithelium, so just the epithelial cells that get scraped off as chyme is moving through, the cells themselves will rupture and release more digestive enzymes into your chyme that will help break down your food. So this is actually a really complex area of function, and So just kind of keep in mind, we're just hitting the high points. We're hitting the major enzymes. But that with future research, this list could be expanded someday in our 2-semester A&P books. Now, the small intestine does have a lot of movement. It does do segmentation and peristalsis. So remember, segmentation is going to help to mix chyme with all the different secretions coming in. The constant churning of chyme is going to bring the nutrients into contact with your mucosa with that brush border to help finish digestion and then absorb the nutrients. And the peristalsis is going to keep moving your chyme through the duodenum, the first portion of the small intestine, through the jejunum, which is the second portion, and then into the ileum. And by the time you get to the ileum and your absorption is all complete, it helps to move the remaining residue into your large intestine. You also have what is called a gastroileal reflex. This reflex is triggered by having food in the stomach. And what this reflex will do is it's going to enhance segmentation. So it basically tells the small intestine, you need to hurry up digestion down there. We've got new food in the stomach waiting to come in. And enhancing segmentation is going to enhance your ability to break down your food and get those nutrients absorbed. It also opens the ileal orifice so that you can start emptying the contents of the small intestine into the large intestine, which you need to do because you have to empty the small to make room for the new food coming in through the stomach.